like eukaryotic, cell wall, and plasma membrane. We studied about genetic structure of a prokaryotic cell, also plasmid DNA, which is an additional DNA, sorry, plasmid DNA, which is an additional DNA which is present in the prokaryotic cell. We also studied about different sizes, different types of cells, which is we did not just study it for prokaryotic, okay. And today we are going to study about eukaryotic cell. So if you don't know, Ayushman. I am from India and I am your IMAT educator. Today we are going to study about eukaryotic cell and what organisms are made of eukaryotic cell. Generally, all the multicellular organism, also like some unicellular organism as well, which are from kingdom Protista. And the eukaryotes, they include all the protists, as I just told you, or multicellular organisms like plants, animals, and fungi. So eukaryotic cell, only single cellular organism they make are the protists, except that plants, animals, and fungi, all of them are multicellular organism. And in eukaryotic cell, there is extensive compartmentalization. What do we mean by extensive compartmentalization of cytoplasm? So, as we know that in a eukaryotic cell, there is a presence of some things which has a nature which is the nature of being membrane bound. In prokaryotes, there are not their membranes are not present on every single organelle, but in case of eukaryotic cell, there are organelles which are surrounded by membranes. Like we talk about endoplasmic reticulum, like Golgi complex, like lysosomes, okay, like chloroplast. So yeah, so extensive compartmentalization. So like let me just show you. So it is a eukaryotic cell, and if you look at that, then it is a plant's eukaryotic cell. Okay, so here you can see that there is a organelle, and also this big vacuole, it is also organelle. So there are different sort of compartment, different types of compartment in the cytoplasm, which gives rise to the statement extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. And the eukaryotic cell, they possess an organized nucleus with a nuclear envelope. In addition, nuclear in addition, eukaryotic cell, they also have a variety of complex locomotor cytoskeletal structure. So, as we know that in prokaryotes there is no well-defined nucleus, the genetic material is in the cytoplasm with no membrane around it. But in case of eukaryotes, there is genetic material is inside the nucleus. Okay, genetic material is inside the nucleus. Okay, inside the nucleus. But in prokaryotes, it is not the same. Also, mm, I don't know. I mean, should I use this because it looks more beautiful? Okay, just leave it. Okay, we'll study for it. And in addition, eukaryotic cells have a variety of complex locomotor and skeletal structure. So when we studied about prokaryotic cell, in prokaryotic cell, if the cell is motile, then it must have flagella. Okay, but in eukaryotic cell there are different types of locomotory and movement structure, which are also made of cytoskeletal structure, which we'll study further in the upcoming lectures. And when you talk about motility in a eukaryotic cell, it is done by flagella, whose structure is different from the from the prokaryotic flagella, and also we use cilia for the movement. Okay, Their genetic material is organized into the chromosome. The genetic material of a eukaryotic cell is organized in the form of chromosome. But if you like want to see the chromosome, then you can only see them during the metaphase phase of the cell cycle. Cell cycle will be our next topic after that chapter, not after that lecture. Um, all the eukaryotic cell they are not identical. Plant and animal cell are different as Phono possesses cell wall, plastid, and a large central vacuole, which are absent in animal cells. So, every I think I should keep this. Yeah, I should keep it clean. Okay. Hmm. So here, um, all the eukaryotic cells, all the eukaryotic cells are not identical. Eukaryotic cells they are different. Okay, and like if you look at a protista, it has it is a different kind of cell as compared to a plant cell, as compared to a animal cell. Okay, in case of protista cell, they generally resembles to both animal and plant cells. Okay, and when you look at the animal and plant cell, in plant cell you found like the things we just studied, a large central vacuole, and also what you find cell wall, you find plasmodesmata connections, you find chloroplast or we say plastids, right? But in animal cell you don't find them. But in animal cell you find a locomotive structure centriole, or we studied it yesterday, centrosome, which you don't find in the plant cell. Okay. Like we here it is just written here that animal cells have centrioles which are absent in almost all of the plant cells. Okay. And let me just mm, actually I just wanna keep it faster, right? Okay, so now we are going to study about these diagrams, and there are two diagrams, one being of a plant cell and the another one being of a animal cell, right? So um, yeah, first we'll take a look at the plant cell. So we will go from inside to outside regions. Okay, so inside here is a nucleus. This thing is this is called the nucleus, and let me use this and this is the nucleus. Okay, and inside the nucleus there's a separate region which do not have any membrane. Okay, it do not have any membrane, which is called nucleolus. In nucleolus there's a rRNA synthesis which occurs, and when you look at the nuclear membrane, like this is the nuclear membrane, you look at these small pores, you look at these small pores, they are called nuclear pores. They, and what does it do? They help in the transport of material from inside and outside the nucleus into the cytoplasm or from the cytoplasm. Okay, and from the outer membrane of the nucleus there are these organelles coming out which are called endoplasmic reticulum. Okay, like it should be mentioned somewhere here. Yeah, this one is the endoplasmic reticulum. What is the meaning of rough endoplasmic reticulum? I will tell you about that. Okay, and after that here comes these lysosomes. And who makes the lysosome? Lysosomes are made by the help of Golgi apparatus. Okay, Some materials will go from endoplasmic reticulum to Golgi apparatus. Golgi apparatus will process it and form lysosome, and it forms a lot more other materials as well, not just lysosomes. Okay, and here is the mitochondria. Mitochondria, which you all guys know, the powerhouse of the cell, and here these are the chloroplasts which help in the synthesis of glucose, or we say which help in the which helps in the process of photosynthesis. And here is the large central vacuole. Okay, and when you come at the edge of the cell, you see that here, like, if there are thin layer, there is a very thin layer which is the plasma membrane, which you can't see in the diagram, but I'm telling you that, look, plasma membrane, this line is the plasma membrane. Okay, and then there's a thick membrane like this one, it is the cell wall. Okay, and in case of cell wall, there you'll see that there are two cell walls, so it is the adjacent, it is the cell wall of the adjacent cell. And when you look at this space, which connects the cytoplasm of one cell to the cytoplasm of another cell, in this space is known as plasmodesmata, which helps in connecting the space of cytoplasm of one cell to the another adjacent cell in a plant cell. Okay, in plants, not in a plant cell. After that, here comes our beloved animal cell. Okay, it is the animal cell. In animal cell, we will start again from the inside. Inside here is the nucleus. Okay, yeah, here is the nucleus, and here it is the nucleolus. Okay, and there are those pores, nuclear pores, and the membrane. Uh, it is a membrane which has pores in it. Okay, also it is called perinuclear membrane. We'll study about it later. And with attached to the membrane, there's the endoplasmic reticulum, and it is the rough one because it has these dots which are ribosomes. And also this one is the smooth endoplasmic reticulum because it do not have these little dots which are the ribosomes. Okay, after that, here comes the Golgi apparatus, which helps in forming some substances like lysosome, our favorite. And there are peroxisome which are also I think, yeah, which are also present only in the animal cells. And here is the mitochondria. Mitochondria, it's, as you know, the powerhouse of an cell. And then here comes the plasma membrane. Okay, here you can see that there are no cell wall. The plasma membrane, it is just a normal plasma membrane, it do not have a cell wall. Okay, like, yeah, plasma membrane, this, it do not have a cell wall. And here like structure, these are the microvilli, and the use of microvilli is generally absorption. Right, so now we will start the cell wall. Just let me get some water and check if the stream is working or anyone came on the stream. Mhm. Okay, let me just drink some water. Mhm. Okay, so let me just turn on the do not disturb button. I don't want this notification in my class. Where like it is, let's find it with me. Hmm. Yeah, that's it. Yeah, do not disturb. Yeah. Okay. So now we will study cell membrane. Cell membrane is a very interesting topic. So generally, the detailed structure of every organ of a cell was discovered after the electron microscopes invention. Okay, but still there were some chemical studies which were done on the human RBC to predict the structure of the cell membrane. Okay, so when those chemical studies were done, there were some results which we got. Those studies showed that cell membrane is mainly composed of lipid and proteins. The chemical studies, they showed that the cell membrane was generally made of what? Lipids and proteins. The major lipids were phospholipids that were arranged in a bilayer. Okay, it means that the lipids, they were arranged in a bilayer, and the type of lipids they were the phospholipids. Yep. Also, the lipids were arranged within the membrane with the polar head towards the outer side and the head of oblique tail towards the inner part. Okay. So, I think, yeah. So when you look at that membrane, what it says that the phospholipids have two parts. They had a head and they had tail. Okay, the head was polar. What do you mean by polar? The one which can be mixed in the water, which has some charge, and their tail was non-polar, which do not have charge and which cannot even get charged. Okay, so it will not interact with water. So the polar head in that membrane structure stays outward. Okay, and it was a bilayer, so on the other side also the polar head is outward, the tail was inward. What is the efficient, what do we get after that? So what do we get because of that? uh, what we get is that the non-polar tail stays inside, and because of its non-polar nature and outside there's polar nature, the non-polar nature of the tail, it can be easily protected, and the polar heads outside, they can easily interact with the environment. Okay, and this ensures that the non-polar part of the saturated hydrocarbon, the non-polar part is made of saturated hydrocarbon, is protected from the aqueous environment. And in addition to phospholipid, membrane also had cholesterols. Okay, in the membrane, in addition to phospholipids, they also had cholesterol. Later, biochemical investigations clearly revealed that the cell membranes also possess proteins and carbohydrates. Later, it was also known that on the membrane there were proteins, also carbohydrates, and sometimes when proteins and carbohydrates were together, they were also called glycoproteins. Okay, and the ratio the ratio of protein and lipid varies considerably in different cell types. The ratio of protein and lipids, which vary considerably in different cell types. But in case of human cell, it was generally 52% of protein as per 40% of lipids. Okay. And depending on the age of extraction of the membranous proteins, there were two types of proteins. First one being integra and second one being peripheral. The integral proteins were sometimes completely, or I should improve the handwriting. I mean, that's yeah. So the membranal proteins were the first one integral, and the second one being um, partially peripheral. No, the second one was, sorry, my bad, peripheral. Okay. So this was all about proteins and cell membrane. Not all. There's some more, and also one thing which I want to tell you, that generally the integral proteins, um, like these ones, their function is to transport the materials from outside to inside of the cell and inside to outside of the cell.
दूसरा और भी कई चीज़ें जिसमें कि